We did an experiment to test our hypothesis. If you grow bacteria in a medium with an antibiotic, then after several generations, all the bacteria will be resistant to the antibiotic. We washed our hands before and after our work.
I always tied my hair back before doing any lab work, and we all wore lab coats.
We wore goggles too, and disinfected the benchtop before and after any lab work.
On the first day, Amber got our parental culture of Pseudomonas fluorescens. I used it to inoculate 2 new cultures. I labeled a test tube of nutrient broth A, and a test tube of nutrient broth with the antibiotic kanamycin B. I lit the Bunsen burner so that I could flame the open mouths of the culture tubes to prevent them from getting contaminated. I opened a sterile pipette package enough to insert it into a pipette pump. Then I opened the parental culture tube. I flamed its open mouth and removed 1/10 of a milliliter of the parental culture. I flamed the mouth of the test tube again, and then replaced the cap. Next, I opened culture tube A and inoculated the medium in the test tube with the sample I had removed from the parental culture. I flamed the mouth of the tube and replaced the cap. I put the pipette in a beaker of disinfectant. I repeated the procedure for culture tube B. I inoculated it with 1/10 of a milliliter from the parental culture, following the same sterile technique procedures, such as flaming the mouths of the test tubes and putting the used pipette in disinfectant.
In order to see whether any bacteria in the parental culture were resistant to kanamycin, we plated samples of it on nutrient agar with and without kanamycin. I labeled a plate with just nutrient agar 1 and a plate with nutrient agar containing kanamycin 2. I used a pipetter with a sterile pipette tip to remove 1/10 of a milliliter of the parental culture. I flamed the mouth of the test tube before and after removing the sample from the culture. Then I deposited the sample of the culture in the middle of plate 1. I sterilized a glass rod spreader by dipping it in alcohol and then touching it to the flame of the Bunsen burner. After the alcohol had been burned off, I cooled it briefly and then used it to spread the drop of culture evenly over plate 1. I repeated the procedure to make plate 2. After I deposited the sample in the middle of the plate, I sterilized the spreader by dipping it in alcohol and burning off the alcohol. Then I used it to spread the culture evenly over plate 2. We put the culture tubes and the plates into a 25-degree incubator.